Okay, we're nearly there. We're into the final session. I'm waiting for the boss, wherever the boss is. Boss is here, right? Great. So it's really great. We're into the final kind of session with two talks, and this is going to be two talks on disruptive sort of technologies or new technologies, which could be very, very exciting and interesting. And it's a really great pleasure to introduce Steve Shi from Concordia, who's Associate Professor in Electrical and Computer Engineering, and also is a collaborator with Vince. And Steve's going to tell us about how he's going to get rid of all of these mainframe biofoundries and make everything really, really small. So a miniaturized biofoundries using microfluidics. So over to you, Steve.
Thanks so much, Paul, and appreciate the pressure too. Yeah. But just want to say thanks a lot for inviting me here to speak. I traveled very far, 10 minutes down the street. Yeah, I'm going to sort of give you guys sort of overview what my lab has been working on in terms of miniaturized biofoundries and using microfluidics. So I'm sure everyone knows what we're all doing here in terms of synthetic biology, trying to create and assemble a bunch of genetic materials into cells. And we heard two great talks just previous to this. We've been really focused on this whole engineering cycle of design, build, test, and learn, and trying to figure out, how we can solve some of these challenges related to this, and specifically challenges related to standardization, reproducibility, and automation, which I'm sure many of you here have been sort of experiencing and trying to solve for yourselves. I guess for us, I worked with robotics a little bit when I was doing my PhD, but I sort of fell in love with this sort of miniaturized technologies using microfluidics. And in fact, my lab has been focused on trying to expedite or automate this whole cycle, or we think we can complete the loop using these droplet-based technologies. And what we'll focus on, in fact, is not so much on the one on the top there, but sort of on the bottom right, where we're using digital microfluidics. So I don't know, I'm not sure if everyone knows everything about digital microfluidics, but essentially here we don't have any pumps or valves or channels, if you ever worked with microfluidics before. But what we're doing here is we're manipulating droplets on an array of electrodes. So just by doing this, we can essentially perform a number of different droplet operations. We can dispense, we can move, merge, mix, split, in sort of any order in any fashion you like. And the way that this whole thing sort of happens, or this magic happens, I would say, is essentially that you apply a potential to an electrode. We have a sort of this insulating layer here where it stores charge. You can think of it as like some sort of capacitor. And then because this sort of stored charge, you can then have these opposite charges sort of form on the edge of the droplet, and sort of acts as like a handle to pull the droplet to the activated one. And I always give this example where, at least for my kids, they're always so excited when I put this balloon on my hair and then it sticks on the wall. This is sort of the same idea where, have the static comb, and then if you put it near this water, sort of bends towards the static comb. Okay, and using this technology, I think there are a number of lots of advantages, and one of them I'd like to highlight here is obviously automation is one of them. Here, the whole reason why we love this technology is that you can have this programmable approach towards synthetic biology. You can upload your sort of device design that's directly on the screen. And each of these little boxes here represents an electrode. So you can kind of have like your iPad, you can like program your sort of path on how you manipulate droplets. And this all happens with, Arduinos and a few switches. And this can be sort of on your benchtop. We have this right in our lab, and we're really hoping that other people can possibly adapt or adopt, I should say, some of these technologies in their own laboratories. One common question I always get is, well, how do you fabricate these devices? Do I have to go to the clean room and all that? Well, the other advantage with digital microfluidics is that you can do this again in your own laboratory, just with a printer and a few copper boards. So all you have to do is you make a design on your favorite CAD software. You can print this on paper, you transfer the ink, then you perform some etching, and then you can start plugging away into your automation system and perform all the droplets that you want. And these copper boards are quite inexpensive, maybe I guess maybe pre-inflation, but so far right now, I think we're looking at about $2 to $3 per copper board. So with this advantage on automation and being able to program some of the biology and then be able to print some of these devices in the lab, one of my students has sort of took the challenge to try to automate the build step. Basically to, take these DNA parts, use our favorite assembly method, create these plasmids, and then be able to transform and then select the ones that sort of been transformed and has our plasmid part. And then we would do everything this process here directly on the device. And so he came up with a method or a device where you have these sort of linear base electrodes here, basically reservoirs to store our DNA parts, our favorite enzyme, and then chemically competent cells so that we can perform the transformation. The other advantage with using digital microfluidics is that we can also integrate other components to the device. So here, because we're using heat shock and using thermal cycling conditions, we can integrate this Peltier heater directly below our device, so we can have like these heating regions for heat shock. So as everyone knows, you have 37, 42 degrees, as well as 4 degrees. And again, all these devices that fits right in the palm of your hand. Reagent volume is around 1 microliter, but you can even go lower than that. And again, it's easy integration with other components that you want with this system. Now, my student at the time, he was like, well, I want something that I can easily program, something that I can standardize, and something that can be easily copied. And so he did design this way of linearization on the device. And the whole reason for that is that you can easily copy these blocks and then be able to program them easily on your sort of software. So here, let's say you have your reservoirs and then you have these moving electrodes, then you can just take this and copy, several n times and then easily do this for the number of different, depending on the number of reagents you have, taking these block units and copy them and pasting them very easily. And then by doing that for the programming side, you can then sort of say, well, I want to dispense some of these guys to this block number. I can move droplets to some other block number, or if you want to deliver specific droplets to other sites on the device too. And again, all programmed by this block number. So it's really easy to make, easy to program, and again, very easy to design. But he sort of, I guess, ran into the challenge very quickly in terms of it's not sort of an easy translation. It's not like you just take your favorite assembly method, you just reduce the volumes, and then boom, voila, you have your assembled product. In fact, in initial tests, plasmids were not assembly, obviously, and then low transformation efficiency. We had a lot of sample evaporation, and then the droplets were very hard to move. And so a lot of times what we're trying to do is trying to solve some of these challenges. And one of the challenges why, I guess if we're looking at droplets or why they were hard to move, one thing I didn't tell you before is that while you're going through this process here of sort of your do-it-yourself microfluidic devices, a lot of these, platforms that we or these devices that we make ourselves are these square-based electrodes. And when we actually fabricate them, we have these very large gaps. So you can imagine if you have these very large gaps between electrodes, droplets will be harder to move because you're applying these electrical fields, and then the droplet's not experiencing enough force to move the droplet to the activated one. So this was a problem. We couldn't really use, the typical standard way for electrode designs, but my student did come up with a nice method where we have these sort of slash-based electrodes. And by doing that, we can actually move them very easily on the device and still maintain a very nice driving force for the droplets. And the idea is that here you sort of have this schematic here. And basically, again, we follow the same sort of sequence on turning on and off the electrodes. But the nice thing is that now droplets are sort of just touching the adjacent electrode, which really helps the droplet to move to the adjacent one. So this is really great. We're able to move the droplets now, but then we would start to face another challenge, which I sort of mentioned before, is this sample evaporation. Over time, if you're definitely trying to thermal cycle, which is more or what people have been experiencing, or if you use any sort of assembly method, you need some sort of thermal cycling. And as you sort of saw in this video, a lot of these droplets start to evaporate very quickly. And what we found is that just by evaporation, we're increasing the concentration of these assembled products, which again reduce some of the transformation efficiency at the end. So what do we do next? But my student came up again with a nice, clever idea where we can replenish the volumes right away. And one of the advantages I said with digital microfluidics is that you can integrate different components directly on or below the device. And so here what we're doing is, as you can see from the video, is that if it reaches below some sort of volume, it would activate the water reservoir as you shown here, and then it would dispense one and replenish the sample reservoir directly. And how we do that essentially is that because we have this insulating layer that's that is sitting on top of our electrode, it's basically used as a way to sense if the droplet is reducing in volume or not. And so here, as you can sort of just see, this is the sample electrode. It sort of goes below some sort of volume now. This triggers the droplet dispensing from the water reservoir, and then we start to dispense, and then this brings it to the sample electrode. Now, if the water reservoir goes below some sort of threshold as well, we sort of 3D printed this syringe pump that directly feeds into the water reservoir, so that we also sort of replenish the water reservoir there. And so we did some few tests. Things look very promising. We actually assembled this 3 kilobase plasmid directly on the device, tried a number of different cycles, tried it on the bench top, compared it to, no replenishment with replenishment. And right away, you can see that, if you look at the transformation efficiencies, these were with replenishment. And on the device, we actually saw higher efficiencies there. So this is great. We also came up actually with another method where instead of replenishment, why not just store more volume directly on the reservoir? And so what we did was we created this sort of well that sits on top of our actual electrode reservoir. And by doing that, we can just have more volume. This way you don't need to refill. And then we can have almost unlimited number of dispensing, or at least for the applications of assembly and transformation, we can have a limited number of droplet dispensing steps. And as I was saying before, we can also integrate heating right below. My student here, Asan, was able to create this sort of feedback system with heating as well, because what he observed was that without any feedback, it's very hard to control the temperature at a constant value. And so just from just using this reservoir system with some feedback on the temperature right away, just from, again, just taking some transformation experiments with single plasmids, we already could see that with this sort of temperature control with our well, we get better efficiencies when we don't have such things on the device. And then we also did sort of a co-transformation experiment where you can transform two different types of plasma at the same time. And again, we would achieve something very similar as well in terms of, having the temperature control with the well. So this worked, or these systems that with the replenishment and with this whole reservoir system, it worked really well for very small plasmids. So we're looking at 3 kilobases. But if you want to, work towards, some major application in terms of, let's say, gene editing, you're looking at plasmids that are quite large in the 10 kilobases. And we started having, again, some trouble with sort of the transformation on those sides or that kind of cargo for our work. And so my student actually looked into the actual temperature scheme that's happening on the device. Heat transfer is much different on the miniaturized scale compared to the macro scale. And this is sort of what he plotted, what he took one of his favorite thermal cyclers in the lab. And what he did was he put a sensor just directly in the tube. And then he tried to do the heat shock and measure the sort of heating curve. And what he observed was very interesting is that, you sort of see this ramp up, you do this heat shock for like 30 seconds, and then you see this ramp down to 0. But what we noticed right away is that it's not exactly what we envisioned, right? It's not like we go right up to 42 in like a span of milliseconds and then stays at 42. It actually has a slow ramp up. And so we're like thinking, well, that's maybe something that we need to mimic on the device. We actually looked into that, where we looked at these different time constants, which means that, how quickly the temperature can ramp up to our favorite temperature. In fact, that had a huge effect on the transformation efficiency. So the benchtop, we measured, this time constant to be 17, which is shown by the blue here. But then when we start to change that or mimic that on our device, which is shown in this light green one, we actually get somewhat of a different transformation efficiency on the device. And in fact, what we found was that 42 degrees was not actually the ideal for this process that we're using on the device. And then if we change this to even faster time constants like 0.5, as shown by this gold line here, which we actually reach 42, our efficiencies actually dropped significantly. And so we said, okay, well, maybe we should change the temperature altogether, but using the same time constant. And in fact, we looked at 37 degrees. And now what we're seeing is that, with different time constants, the one with the 0.5 or quicker ramp up, we actually get better transformation efficiency, or the best that we've seen so far on the device. So I guess the moral of the story here is that, when we're trying to do something in the macro scale, you can't always translate to the micro scale directly, just because, different forces or different phenomena like heat transfer are much different on the micro scale. But with this now we're able to have this replenishment system. We're able to sort of monitor this heating curve. This actually led us to, perform like sort of a model experiment where we could just assemble, different parts and see if we actually get any results or increase the fidelity on these transformation efficiency. And so we use this violacein model, which I'm sure many of you know. It has like 5 gene parts. And what we found here was the following. The main key data that we got was that now we're able to get these very nice efficiencies, at least very comparable to the bench scale. Before, when we didn't have any of these sort of integration with heating and then with the lack of replenishment, we essentially saw 0. But now we're able to get some of these colonies, really pretty good efficiencies. And then we're actually sequence some of these, and then we're able to get the right size and the right parts and the right sequences as well. And we did it for, the 3 kilobase plasmids as well, as I sort of discussed before. And again, we're getting the efficiencies that we've seen before. So this is what sort of our current efforts right now in terms of assembling and transformation. And we're also thinking on sort of working towards, how to increase throughput. And then as well, at the same time, we're also looking into DNA writing. We're still working on this right now. But what I could say right now is that, we're using these digital microfluidic platforms for phosphoramidite chemistry. We're using these MENAC beads to grow our strands. And just some preliminary work that we show here is that, just synthesizing, a few T's or four temers and 5 and 6. We're actually able to get these strands and off the chip very, very easily. So I guess in terms of time, I guess, I'll sort of close it up here. But, we sort of wrote this review or this book chapter that discusses, how we can integrate microfluidics within synthetic biology. So if you're interested to know more, I can send you sort of this book chapter that we wrote. I know there's been a lot of work with, or a lot of interest, especially from the last session in gene editing, and our lab has been tackling a lot of these automation issues with gene editing or building plasmids for gene editing or transfection. And so my lab has published some work in terms of lipid-based transfection, viral-based work. We have this tri-droplet system that we just submitted. And then we also looked at the very challenging, as sort of Kevin's alluded to, and sort of the single cell selection and cloning, which we also have published. And from that, I just like to say, a lot of the work that I've shown here are from amazing set of students. Definitely like to thank James here, who's nearing the end of his PhD, which I'm very sad about, but he did a lot of the work here. My alumni student, Ehsan, and then Brian, who's sort of spearheading the work with DNA synthesis. And of course, I'd like to thank my funding sources, and of course, I'd like to take any questions that you may have.
Thanks very much, Steve. I think we can take 1 or 2 questions. I think we're okay for that. If anyone's got any questions.
Hi there. Thanks for the nice talk.
Thank you.
So I'm curious, it seems like there's been a couple of commercial kind of electrowetting solutions, like the NeoPrep from Illumina that came out like, I don't know, 5 or so years ago. So do you have any, I guess, speculation as for why there hasn't been like a commercial electrowetting kind of success as of now, I guess?
Sure. Yeah, so the NeoPrep system now with Illumina, I mean, as I've seen right now, they don't have actual product that's out. I think that's closed down. There is a company called Miraculous, which is also not in San Diego, but in San Francisco. They do have a sequencing platform, and I think they have one here in Montreal, not in my lab, but over at McGill. So it is out. I just think with digital microfluidics, there's it's a lot of inherent challenges that, as I sort of mentioned, with electrodes, with evaporation. One that I didn't mention too much is, something called fouling. So the droplets that you place on these devices, it will just foul the surface, and fouling surface will prevent you to move any droplets further down the line. That's a big issue with a lot of the sequencing prep or sample prep because of a lot of the liquids that they use is very viscous. So I think that's sort of the biggest challenge that I've seen so far in digital microfluidics. I see other challenges sort of more and more being solved, but this one, the fouling one is one that's sort of preventing, I would say, into the market.
Okay. Great. Well, thanks, Steve. We should probably move on.
Sure.
Yeah. It's not very often you get to introduce one of your PhD students, but anyway, here I am. Kiba is Imperial through and through, undergraduate Imperial, master's Imperial, and just defended her PhD a few weeks ago at Imperial, and is going to tell us about cell-free systems, I hope. So over to you, Kiba.
Thank you. or do I need to plug? Sorry. Sorry, I've got a dodgy thumb. Thanks. Thanks. Okay, thank you. Thanks, Paul. Yeah, so as Paul said, I'm Kiva Canavan. I just finished my PhD at Imperial. And yeah, my PhD was on the development and optimization of mammalian cell-free systems. So we talked a bit this weekend about the role of biofoundries, one of which being teaching students and educating students. So I guess I'm kind of an example of that, and because I got to use some of the automation equipment at the London Biofoundry. Yeah, so I'm going to talk a bit about mammalian cell-free systems and try and focus it towards automation and hopefully not drift too far away from that topic. Yeah, so cell-free systems, for those that aren't aware, I guess, cell-free is a protein. Cell-free protein synthesis is an in vitro tool which uses the protein-making machinery of a cell outside of the constraints of a living system. So the type of cell-free systems I used are lysate-based. So you take a cell, break it open, remove the cellular debris, and you're left with cellular lysate that contains the protein-making machinery necessary for protein synthesis. Because you've broken open the cell, it's no longer living. Obviously, it's no longer able to regenerate certain components such as energy, tRNAs, NTPs, etc. So these are supplemented in when running a cell-free reaction, and you add in your DNA or mRNA of interest that expresses your protein of interest. Incubate the reaction for generally 4 to 10 hours, or you can run it for longer using dialysis to extend runtime, so up to 36, 48 hours, and produce your protein of interest. So there's many advantages of cell-free systems. One being that they're non-living, so you're able to produce proteins that might be toxic to whole cells because you don't have that requirement to maintain cell viability. So one such example is producing membrane proteins, which can often disrupt membrane and cause cell lysis. This can be done in cell-free without that toxic effect, which is quite important as a biotherapeutic sort of application. It's an open system, so it's easy to manipulate. So if you wanted to say add in certain chaperone protein to help protein folding, rather than say having to engineer a cell line, which is obviously a very non-trivial task, as Syntheco quite clearly showed. Instead you can just add it in as another reaction component. It's a rapid process, so you can get results within a matter of hours, and it's ideal for high throughput experimentation, which hopefully I'll be able to show. There's some limitations. You generally get lower yields than whole cells, but it depends on sort of the reactions that you're running. And I think over the past few years, the yields have definitely increased significantly. And batch-to-batch variability is another limitation, which I was trying to investigate and overcome in my PhD. Because obviously synthetic biology, we're trying to engineer biology, we want predictable, reproducible outputs. So batch-to-batch variability is a big limitation to that. So if we can overcome that, then hopefully it can be a better tool for the future. Some of the applications, there's many applications. Some of the applications in our lab include biosensors. So Lauren was a PhD student for a few years ago who developed the cystic fibrosis biosensor to detect pathogens in sputum samples from cystic fibrosis patients. Other applications include protein synthesis, as mentioned, protein engineering using non-conical amino acids, using pure-based cell-free. It can be used as a sort of bottom-up approach to synthetic cells. But the biggest application that probably has the most relevance to automation is prototyping. Because we can do high throughput screening, we can test hundreds of constructs in a very short time frame. So to pick out the best constructs and then before scaling up or downstream applications. So there's a variety of commercial cell-free systems exist. So NEB sell the E. coli cell-free system. Thermo Fisher sell a HeLa cell-free system. And there's many other examples. They work very well, but they're limited partly because they're very expensive. So yeah, limits applications. And also there's a variety of different chassis that are used, but not all of them exist. Yeah, and also because they're sort of pre-mixed, you as a user don't have ultimate control over the contents. So that's why we focused on developing our own, specifically HEC-293 cell-free, because there's no commercial cell-free system from that cell type, and there was no published papers on HEC-293 cell-free for synthetic biology applications. So most cell-free systems that you probably hear about are bacterial based, because they're simpler, easy to produce. But over the last few years, there's been increasing research into mammalian cell-free. Mammalian cell-free obviously has the added complexity of compartmentalization. So the idea behind cell-free being everything is open without membrane barriers. The fact that there's lots of compartmentalization of mammalian cells is obviously a complication, but the way the biggest complication being the presence of a nucleus. So the way we overcome this is the nucleus is removed, and you instead use supplement T7 polymerase to enable transcription and an IRS element to enable translation without the requirement for capping. This works very well. It's been done in most eukaryotic cell-free systems, and enables high yields. So that limitation has been overcome. And the reason we use mammalian cell-free is because you have the added capacity of producing complex proteins that require post-translational modifications, such as antibodies. The second big application is prototyping. So obviously, if you want to screen many constructs and you want to do it in cell-free prior to potentially expressing it in whole cells, you obviously want a cell-free system of a corresponding type. So hence, if you want to express something in HeLa or HEC cells, you'd want a HeLa or HEC cell-free system. So how was automation used with cell-free? So as explained, it's an open system. And the way that we set it up is using the acoustic liquid handling platform. So this uses sound waves to produce droplets to transfer liquid from a source plate to a destination plate. And it works very well. You just alter the settings depending on which components you are adding in. So lysate has a slightly different viscosity to, say, your DNA or other components. So you program the echo to transfer in the right conditions. You have, obviously, it gives you significant control over what you want to add. So you can automate the robot to add in different components as necessary, and then incubate the reaction. And generally, the readout we've been using is GFP or aptamers, or some sort of fluorescence assay. So you can gain time course measurements using the acoustic liquid handling platform, which has the plate reader, sorry, I couldn't remember the word, plate reader attached all on one platform at the London Biofoundry. Yeah, so we use GFP mostly to detect expression, and we produce the malachite green aptamer so that we can detect transcription and translation from the same construct, which helped us during the sort of optimization stages to be able to identify when there's bottlenecks, to identify if it's in transcription or it's translation, or if the whole system's just completely crashed and not working. Yeah, and as shown, you can get mRNA measurements over 10 hours and lower graphs as GFP production. Yeah, so when producing cell-free extracts, I did a lot of optimization of many different processes. You don't need to take too much from this, other than the fact that there's significant variability, and it wasn't always easy to be able to identify a clear optimum condition. So in some cases, you can say that, harvesting at 48 hours seemed to be clearly the optimum, although there's some leeway. And pH 7.5 was the best pH buffer. But when looking at timing and pressures, obviously the fact that there's significant variation in lysates made it very difficult to draw conclusions. So we used DOE to sort of measure all of the different input factors, whether they could be controlled or not be controlled, to try and identify where this variation comes in, to then in turn minimize said variation. And what we basically found from that is we found the optimum pH, but basically the starting batch of lysate was actually the most significant in determining the output of activity of the lysate, which given all of the different parameters I looked at, and the order and the temperature and everything that I thought might have an effect, the fact that it was kind of outside of my control was interesting. But yes, so then that kind of highlighted the fact that there's probably needs more control, because at the time I was just growing cells, suspension cells manually, but it kind of highlighted the need for manual cell culture or bioreactors, etc., to be able to ensure that cell growth is maintained, and that those differences don't remain significant. So I know at the Biofoundry now we have the Amber 15 for automated cell culture. I unfortunately haven't had the opportunity to be able to use it in my PhD. COVID, etc., everything, things that I wanted to do weren't necessarily able to occur, but hopefully being able to control it's able to measure and control pH to ensure that and cell density to ensure that the cells are grown continuously. So hopefully that will minimize that sort of variation, so then we can downstream better predict and have extract performance that's more reliable. So some of the other things I was also looking at is just when I was setting up reactions is something that I don't know if anyone works with cell-free, but you'll notice that if you use, if you sort of pipette, when you're setting up reactions, if you're pipetting your lysate up and down, you'll get significantly less activity, which I guess makes sense because there's lots of intracellular compartments that you want to remain intact so that you don't release, say, degradation vesicles, etc., and damage your lysate. So I just wanted to better understand that, and it was suggested that mixing just by like flicking the Eppendorf tube that the lysate's in would be a better method than pipetting, which I think is more widely done now. But I wanted to compare it to using Yeko, and I showed that you get better activity in overall, higher production and less variation when using the Echo setup. Yeah, so once we'd made our lysate, our HEC lysates, and they worked, we were getting the same or if not better activity than the commercial HeLa system. We then used it to screen energy mix designs. So we Investigated 15 different components of 3 concentrations. So obviously doing this manually would have not been possible, and also doing every combination was too many to do them all. So use a DOE approach, and we identified HEPs, magnesium glutamate, and creatine phosphate to be the most significant for optimum yield, and kind of identified that something like spermidine isn't necessary and is somewhat detrimental to activity. Yeah, and we actually identified that if you increase the concentration of HEPs in the commercial cell-free kit, then you actually get higher yield, which is interesting. Yeah, so obviously that wouldn't have been possible without the use of automation. Yeah, so some of the applications that we were looking at were screening. So Margarita, a previous PhD student, was testing different DNA concentrations of 5 different DNA constructs with single or double base mutations to identify, within the T7 polymerase promoter and within the IRS element, just to improve yield. And she did this again using the echo. And yeah, identified improvements. And kind of the next step along was doing that for our most recent application using self-amplifying RNA COVID vaccines. It's obviously very topical. So yeah, obviously we have the capacity to test hundreds of constructs. I should have mentioned, we were able to scale down the reactions to 4 microliters. So generally run in either 10 or 4 microliters. Mostly only use 10 if it's necessary, because you have so many different components, you can't transfer them in less than 25 nanoliters. But yeah, generally in 4 microliter reactions. So to be able to screen it wasn't the problem. And we were able to show that you can test multiple different constructs. So then we wanted, and you're also able to synthesize, different components, VLPs, antibodies, etc., and shown to be expressed in cell-free. So we wanted sort of a newer application. So we were testing, the SARS-CoV-2 self-amplifying RNA vaccine. So obviously we'll know about RNA vaccines. The self-amplifying vaccine has the benefit that if you can give it to patients at a lower concentration and it's able to replicate within the body to minimize cost and increase output, which is obviously necessary for pandemic kits. So the way it works is that NSP proteins are expressed. They form a protein with host proteins, and that activates the subgenomic promoter, which then expresses SARS-CoV-2. So at the moment, we're in the process of trying to make this work in HEC-293 cell-free. We're able to express some of the NSP proteins successfully, we haven't been able to express SARS-CoV-2. So we're kind of just investigating the bottleneck of why subgenomic promoter is not activated. There's many different suggestions why that is, but yeah, it's quite early on in the project. So, but it's just to show the capability of HEC-293 cell-free systems. And finally, we talked a lot about software this week, and obviously it's great having automation equipment, but the ability to actually use and run the equipment is somewhat of a bottleneck. So the way that we were doing it was we were using the company Riffin, which were great. We had worked with them for a few years now, I think. And I was setting up sort of scripts and programs to be able to have like a user interface that was user-friendly. So things that there's multiple different inputs that you want to change each time. So sorry, it's tiny, can't read it. But we've got 2 different types of echo that we'd use, the echo 525, the echo 550. There's different source plate types that you need to pick, liquid classes. There's 6 to 8 liquid classes. All of these need to be changed each time for each component. So having to sort of run that through a Python code is somewhat difficult. And obviously I want the next people to come along to be able to use it. And I myself, I'm a biologist. I'm definitely not an automation expert. So I wanted something that, yeah, was sort of user friendly so that you could have multiple different steps, everything necessary for the energy mix setup and the reaction mix with adding the components in, and then to export a Excel file that could then be directly used with the Echo platform. Only downside is that Riffin is now no longer exists. So if sort of a lesson, and maybe we talked about whether you outsource software or use it, develop your own in-house. So yeah, that's probably a benefit of producing it in-house. But there's lots that we learned from it, and I have all the Excel sheets and understand how it works. So I definitely gained a lot of input and understanding on how to sort of what would be a good interface and how to develop Excel, a library of protocols and Excel files for the next user. Yeah, so I think that's basically everything. So I just want to thank Paul and everyone in the Fremont Group and the London Biofoundry for all their help over my PhD. Any questions?
Thank you, Kiva. I guess we've got time for a very quick question if anyone wants to. Oh, someone's up.
Thank you very much. That was very interesting. I have a few questions on the workflow itself. How do you lyse the cells at first?
I use nitrogen cavitation vessel. Sorry, I should have mentioned that. I did put a diagram, but I didn't mention it.
So you can do it high throughput?
That step, no. So that is the making of the lysate is definitely an area where we could put it, we can have more high throughput. It is quite a good method because you can scale it up to larger volumes in nitrogen cavitation vessel. I think the one I have is like a milliliter to 10 mils or so, but you can do it a lot higher. But of all the methods to make lysate, it was the least manual. Which is why I chose it, but it's definitely an area where automation could benefit, because I know they use the pixel for E. coli cell free, but obviously with you don't want to use sonication when you're lysing mammalian cells.
But at some point you must be getting like a kind of a soup of proteins, cell compartments, and DNA. Do you centrifuge the sample to get?
Yes. After you lyse the cells, you've got all the cell debris and everything. So then you centrifuge and you take the supernatant, and that contains the components.
And are you aware of any way of high throughput centrifugation? Because this is one of the bottlenecks that we are currently struggling at the foundry. That are you aware of anything that could allow the high throughput centrifugation of tubes, Falcon plates, or whatever?
No.
Is anyone aware of anything?
Yeah.
Oh, nice. Yeah. Nice. And you mentioned that you got high yields of proteins, because this system was reminding me a bit, I don't know if you heard of the Promega reticulocyte system. Basically, you put your DNA and you have all the components to produce the protein. Yeah. But you only get like at best 2 nanograms of proteins. In your case, what is the max yield?
I think we were getting up to 800 nanomolar at one point of GFP. But that was in batch reactions. Okay. So you get more if you do dialysis and run it for longer, because that's over 10 hours and it sort of plateaus about 6 hours.
Okay. And my last question is, you get your protein within this soup of components. How do you purify it with a tag? Do you have any way to purify it?
Yeah, so when I was mainly just using purification tags when we're producing things like that. We had so we used the high bit tag to be able to detect using the bioluminescence assay, so you could detect your protein and quantify it. But then downstream we were using just purification, sort of based,
such as a flag or whatever, and
so his tag, etc. Yes.
Yeah, that's very cool. Thank you very much.
Thanks. Thank you.
Great.
Okay.
Sure. Yeah.
Yeah, just a quick one. Thanks so much for this talk. I agree with the DOE experiment. I think that's the right approach. And I guess you found the lysate, the type of lysate. So is that different cell types that you found the significant difference, or is it like the way you compare the lysate?
So they're the same cell lysate type.
Same cell lysate.
Yeah, and I varied different conditions because I was trying to work out the best condition to lyse the cells. But even within the same condition, you'd get variation.
Okay, so even following the same parameters.
Yeah,
you still
yeah.
get
So I would,
variation.
yeah, I take a batch of cells and then split it up into say 4 and then do 4 different conditions from that. And obviously, if I replicate that again, then I would get different variation using the same between the same conditions each time. So it was more of a batch, I think. So whether it's the actual starting batch or the handling of that batch, or so if I'm too harsh, obviously mammalian cells are very sensitive, so it's not necessarily the growth. I think there's many different factors as to what it could have been or what is causing that, but yeah, it's the batch of lysate, the starting cells you use.
And just to follow up on that, I saw on the slide you said PBs. That's surprising. Any reason why that was the case?
I guess it just buffers increases the buffer pH, which you wouldn't think is needed that much because we added quite high concentrations and it significantly improved. But yeah, I mean, the pH of the cell lysate matters a lot. If it gets below 6.3, you just get zero activity. So I guess the increased buffering capacity.
Thank you.
Thanks.
Oh, I think Janet's got.
Oh, sorry.
Hi, I'm Janet Reid, CSIRO. I missed how you're growing adherent culture or is suspension culture?
Suspension, yeah.
Oh, okay, yeah. And do you harvest at a particular like cell density or
Yeah, so I try to passage them so that the cell density is the same, but I was mainly doing timings. So I think I tested a few different timings. I think it was about 48 hours was the optimum. So I measure the cell density and the time. So if obviously when I'm passaging them, I'm hoping that they're going to get to that cell density within that amount of time, but I found quantifying cell density is quite inaccurate, so that it wouldn't necessarily always be the same. But yeah, it would be about, I think we harvested them about 2.2 million cells per milliliter.
how are you measuring density? Just curious.
Using Countess.
Yeah.
And Trifon Blue to detect viability.
Okay.
So yeah, if there's any better methods, I'm all ears.
Oh, I used to use a nuclear counter, but
Okay.
I mean, there's much different.
Yeah.
And you don't use protease inhibitors, obviously, because you need protease in the mix for everything for the actual process.
Some people have used protease inhibitors in cell-free, but I didn't find it was beneficial.
Yeah. Okay.
But there was published post protocol where people did.
Okay. There's sounds like there's like so it's not like a complete rupture of everything. It's rupture of the outside membrane, but then the inner compartment still seems to maintain integrity. So
the idea is that the ER is sort of broken up into microsomes that are still functional, and you can still get those post-translational modifications. So yeah.
Yeah, cool. Okay. I think that was all my questions. Thank you.
Thank
Great
you.
talk. Yep.
Thanks.
Thanks, Kiva. That's great. We should move on, I think.
Yep.
Thank you.